Now I'd like to introduce our second speaker. Dr. Ron Milo received a master's in electrical engineering from Tel Aviv University and a PhD in biological physics from the Weizmann Institute's Feinberg Graduate School before conducting his postdoctoral research at Weizmann as well as at Harvard Medical School. Following his time in Cambridge, Dr. Milo returned to Weizmann in 2008 to join the Department of Plant Scientists. He and his wife have 2 daughters, and for what I've been told, he plays a mean harmonica. Please welcome Dr. Ron Milo.
I didn't know I needed to bring my harmonica with me, so I'll try to entertain you by telling you about the science I'm doing. So, you know, growing up, I was really most fascinated by physics. This was my role model, was a physicist by the name of Richard Feynman, working in Caltech, getting the Nobel Prize. Somebody who was really inspiring both as a person as a scientist. So after doing degrees in physics and math and electrical engineering, I was really, even though I was thinking that I'll continue in physics, I was really blown away when I saw these things. So what we see here are lung cells that we have in our body. And today we're able to take new technologies and be able to image them and watch them doing these amazing things as they're moving about, and I'll be telling you a bit more in a minute. And after seeing this, I couldn't resist the temptation of trying to take my background and my education and try to implement it on some of the riddles that we find when looking at those live phenomena. So this is kind of like what I want to tell you, and a bit about the journey I took in doing that. So when we were looking at these cells, I came to the Weizmann Institute and was also blown away by meeting what would become my mentor, Uri Alon, which just arrived. I guess his lab was just like my lab was just 1 year old. That's about the time when I met him. And looking at these cells, we sort of framed the following question. So the cells that I've just shown you, you see that they're continuously dividing. So these are cancerous cells. And in these cancerous cells, you have the phenomena that in order to try and deal with the disease, you're trying to administer all sorts of drugs as part of chemotherapy. And you'll be applying some kind of drug trying to kill all of the cells. But what you often find is that some of the cells would be able to resist the administration of the drug. Obviously, those cells afterwards would proliferate because they're cancerous cells, and then you would have a relapse of the disease. Now, we were asking the question, what makes one cell being different in its ability to resist the drug than another cell? Even though they might have exactly the same DNA content, they're all coming from the same mother cell, so they're all part of the same family, carrying the same kind of information, but still one of them is able to survive and one does not. So there was a question here about what dictates which cells would survive. In order to try and approach that, we were looking at those kind of technologies that enable us to look at these cells very close hand. So what you see here, each of these is a cell. So this is one cell. Oops. The black dots that you see here are within the nucleus where the DNA is. And you can see these cells moving about. So what you see here, we take pictures every 10 minutes. We follow them for a couple of days under the microscope. And we're able to track them and really learn about each one and try to get an understanding of what it does, how it communicates with its fellow cells. And we're able, because in order to get some insight into its interaction with drugs, we need to look at those major components of the cell called proteins. So these are the main machines that do the functionality of the cell. And we developed, based on all sorts of things that have been done in many different parts around the world, a way in order to look at those proteins in single cells. So what you see here, these are the same cells that we saw before, you can see. But in this case, we're looking just at one specific protein which is inside the nucleus where the DNA resides. And we're able to take these movies, which contain tons of information. There's really huge amounts of information, much more than any computer could hold 10 or 15 years ago. And we want to be able to get a picture of what happened inside each of these cells. So what we did is we developed all sorts of computerized methods in order to learn about each one of the cells, tag them, following them up with image processing, and going from this kind of experiment where you start with until the end of the experiment from one cell that was here, this blue cell down here, you might find that now you have 4 progeny cells, 4 grandchildren cells, and you can see what each one did. And now what we'd like to do is to be able to zoom in into each one of these and be able to see what happens within every cell in terms of its proteins and its functionality. So this is exactly what we're able to do. If you could play this movie, you would find that each of these cells, of the 9 cells here, we're able to go and look at, by the color of them, we can know how many proteins do they have, where are they localized, and what happens to them with time. And this could really give us insight into why is it that they respond differently to the drugs. Because you can get this kind of family picture, if you like. What you see here is as a function of time, so this would be hours, you look at the amount of protein that you have. And you can see the cell growing, and then you can see this downfall. This is the point at which you decided to divide. So now you have, from one mother cell, you now have 2 daughter cells. They would in turn grow themselves, and after a few more hours, you would find there would be another division. Now there would be 4 granddaughter cells, and then 8 granddaughter cells. And you really get a family picture. But what you find in this family picture is that even though they might be carrying with them exactly the same messages and exactly the same DNA as the grandmother cell had, now they're starting to show their own character, just like I find that my 2 daughter cells are very different, even though I thought that I'm growing them in kind of the same conditions, they could be so different in how they behave. So also here you would find that these 2 cells decided to divide much earlier than these 2 cells. Maybe they had a different, the mother was telling them all sorts of different techniques about how to make them have children. So that she was more successful here than this mother in terms of enticing them to have children. But these differences that you were able to look here under the microscope, we find that we could correlate them into how those cells would respond to the different administration of drugs. And this really gives us the ability to look and find targets to what could be good options for therapy in order to deal with those cells that are able to resist the therapy. That was just something that I started together with another graduate student working at the Alon lab during my PhD and later during my postdoc. But actually, when we got to this point, 2 things happened. One is that we found that we needed more and more people and resources. We got a very large support and a donation to do that. And the team that was working on it became this, where you could see Uri Alon, which is my mentor, and some of the people that were involved in this project more and more coming and taking, because there's a lot of challenges trying to face it. But at this point, I also finished my PhD and a period of postdoc. And I went overseas. I came here to Boston to do further research at Harvard Medical School. And there I was looking to see what are the questions that I would like to follow up next. And I felt that the grand challenge that I felt most passionate about pursuing was related to the challenge of sustainability and how can we take some of those tools and approaches that we've been developing in trying to understand life and this thing called systems biology and try to see how they could bear on the grand challenges that we have in terms of enabling us to have 7 billion people on the globe, more and more every day, having a higher standard of living, but then we have a limited amount of area to supply them with food, a limited amount of water in order to grow this, a limited amount of resources for dealing with how we affect our atmosphere. And looking at that from a perspective of this combination of different fields, it seems like a focal point is this process at which we're taking much of the resources in order to do what's known as photosynthesis. This is the main process that really drives the biosphere and really is at the core of everything that we're using later on, be it the wood that makes this nice building or the food that we be getting around lunchtime. So basically, the majority of land area, even though it seems sometimes as if it's only being used for shopping malls, it's actually not being used for that. In most parts of the world, it's almost all of the land area that we're using and taking out of the natural surrounding is in order to grow our food. And there's larger and larger pressures while doing it. Similarly, all the fresh water, and you can feel it very strongly growing up in Israel, but also in other parts of the world, is being used in order to water plants. And then finally, when we're thinking about the emissions that we're doing from our cars, from our power plants, so it turns out that 10 times more, the amount that we're throwing into the atmosphere is really huge. It's 20 gigatons every year. But plants actually take up from the atmosphere 10 times more of that carbon every year. But the thing is, they're in balance. They're taking it out, and then in natural process, it goes back. But it means that every way in which we could understand this process better, in which we could affect this process better, could really have a tremendous effect, both in terms of the land we need to use on the planet, the water resources we need to use, and what happens to our atmosphere, to global climate change, etc. So the focus of the lab that I started a year ago was looking at this marvelous process that makes plants grow and enables both plants and bacteria to take energy in the form of sunlight, as well as carbon from the atmosphere, and do this miraculous transformation where you take these two sort of like physical properties and transform them into the key for life, which is sugars. So sugars is the way for life to have a battery to store energy in the form using carbon, and sugars is both what we eat and what enable us to drive all the processes later on in our body. And so we're looking at the different things that happen. This is a schematic of what's known as a chloroplast. That's a place where you take that energy, store it, and transform it into sugars. Now, there's been a very large effort trying to find out how is this process known as carbon fixation actually happens. And there was a Nobel Prize being given for really delineating all of the steps in this transformation, starting from inorganic carbon and transforming it into a sugar that you can eat, that you can use as fuel for your car. But then we were less successful in trying to see, and this is sort of like the focus of what we're trying to think about now, is what if we could do something in order to be inspired by nature about how to make this process more productive. Are we able to take, for example, by looking at different organisms and the different revolutions that are happening that we're mentioning here in terms of our ability to know the genomes, the DNA of different organisms, being able to look at their content in very sophisticated ways, would we be able to look at that and learn lessons about how we could be more productive in how we use our land resources and how we use the water to be more efficient? So what my lab tries to do is to look at that issue of carbon fixation, and being based on what we see in nature about the different components, try to combine these components together in order to give you new ways in order to do carbon fixation, which could be used as for us in order to take, say, solar power or wind power and transform that into fuel that we could later on use for cars, but do it in a way that we're really inspired by how nature does that, because it had quite a lot of years to study and experiment in how to do it, and that could really give us a lot of hints. So what we're doing is we're developing all sorts of pathways, these things that happen in every cell of our body that enable us to transform. So every circle here would be a carbon atom, and we're transforming into different types and enabling us to close circles where we're fixing carbon from the atmosphere and giving us out products that we are all encoded in the rules of chemistry and can be used later on as energy sources and as food sources. So I'd like to finish up with that, but just mention that if you get to come and visit Weizmann at some point, you can meet some of the people involved in that. So this is the group that we're currently setting up, and I talked about the work of some of them. So this was just when we were hiking in the hills around Jerusalem. So I hope to get and see you either in the hills around Jerusalem or at the Weizmann Institute. Thank you very much.